welcome. So I only discovered Florence and the Machine last year, and it's a bit of a joy to come late to things like that because you've got a great back catalogue to explore. As with Florence and the Machine, I've only really discovered Elizabeth Watkins' work relatively recently, so I've got a big back catalogue to explore. So Liz has come all the way from Perth. She's working at Curtin University, a professor in microbiology, oddly placed in the School of Pharmacy and Biomedical Sciences. So she studies the microbial ecology of environmental systems and covers the fields of mining biotechnology, mineral resource recovery, microbial-induced corrosion, microbial fouling. So her research team's investigating the biotechnological processes for environmental industrial applications, approaches to mitigate biocorrosion and biofouling and bioclogging. But looking at the back catalogue, a lot of the work has been on salt tolerance in a particular genus. So I'm guessing this is your baby, Acidohalobacter. So welcome, Liz, to JAMS annual symposium. Presentation on halotolerant iron and sulfur oxidizing acidophiles with potential for saline water biology. Please take this.
Thank you. And as Mike alluded, is my baby. Let's get the presentation up. And I just wanted to add on to what Emma said about cross-fertilisation within careers. So my career has been very organic, and it is very important I think it is important to have diversity in your background because you can always draw on that. So this is a relatively new field for me. I did do my PhD as a mature age student. I left science for 10 years and then decided science is the best thing to do, so I came back. But I actually came across this field in my role as lecturing microbiology in the molecular genetics and biotechnology degree in the School of Pharmacy and Biomedical Sciences, as you said, very badly placed, and came across Helen Watling from CSIRO. And we discovered very quickly that we could actually form a very strong collaboration, which we did. This is where I started about 10 years ago in this field, and it's developed into, this is my baby, the Acidohalobacter, which are a fascinating genus. So the picture in the background is actually a geothermally heated marine pool in the Aeolian Islands in the Mediterranean. And this is where a number of these organisms have been isolated. They are fascinating in that they're able to tolerate the dual stresses of very low pH and very high salinity. And I'll explain to you over the course of the next 20 minutes or so as to why that's so interesting. So just some background first on biohydrometallurgy or biomining, which is the overall field I'm in. So it's the use of a particular group of organisms, iron and sulfur oxidizing acidophiles, to release metals from their mineral or concentrate. The actual technology that's used depends on the actual metal of interest. And for example, the high net worth metals such as gold and cobalt, they're prepared to put a huge amount of capex into processing those, and so they use bioreactors that are very strictly controlled for temperature, oxygenation, pH, nutrients, etc. Down to, and it literally is done, when you get to the lower net worth minerals such as copper. However, this is an adaptation of a naturally occurring process. These organisms that are used in this process are abundant in areas such as acid mine drainage and acid rock drainage, as shown by these two images here. They are lithotrophs, as I've already said. They oxidize iron and sulfur for their energy source, so the electrons that are released enter the electron transport chain, generating a proton motor force, leading to generation of ATP. They are also acidophiles, as already mentioned. They can grow down as low as pH 1, so all of our culturing is done around about 1.5 to 2.5. And they're chemotrophs, so they fix atmospheric carbon dioxide, and they use the carbon-Bassan-Benson cycle for that. So overall, I think they're a really fascinating group. We grow them on a smell of an oil rag. So if we get contamination, there's actually no excuse for it at all. So the mechanism that they are able to liberate these metals into solution, there are two main metals, and it also depends once again on the mineral of interest, whether that mineral is acid soluble or acid insoluble. But regardless of which it is, the first and most important step is this work, is the bacterial oxidation of ferrous to ferric. So they get the electron out of it, they get their energy, their business is then done. But this generates ferric, and the ferric then acts as an oxidant on the mineral. That leads to the release of the metal into solution, as well as various sulfur moieties. Sulfur oxidizing bacteria will then oxidize that sulfur, once again, for their energy requirements, leading to the release of sulfate and protons. Now, if it's an acid-insoluble mineral, that's the end of the story. If it's an acid-soluble mineral, then the protons will then act further on with metal sulfide, releasing more metal into solution. So underlying mechanism, the bacteria get something out of this. Now, one problem which leads to less than optimal uptake of this technology is the requirement of fresh water for this process. They require vast quantities of fresh water. So the images we have on the left are the Escondida mine in Chile, in the Atacama Desert. Huge operation, it's a BHP Billiton run operation. This heap is 6 kilometres long and 2 kilometres wide, so we're looking at 12 square kilometres of crushed rock. It is what's called a run-of-mine operation. So basically, this is the open cut mine. You can't see, it's not showing up very well in this image, but there are tiny trucks in there. There's huge mining trucks we all know from the northwest. The rock is basically, it's just exploded out of the reserve, it's loaded up onto the trucks, and then it is dumped onto these impermeable membranes, so these huge dumps. It's then irrigated, and I'm in the way of a picture here, it's me visiting the mine. Because we're in the middle of the Atacama Desert, they're actually using a trickle system. Other systems, they use aerial irrigation. So it's irrigated through this trickle system with acidified water, and it's covered by 12 square kilometres of white plastic. It's not very environmentally sound, but it stops evaporation. It then trickles down through the heap. It is aerated, this is an aeration pipe, and then we have the leached copper in solution, and then that's taken away, and the copper is extracted through solvent extraction and electrowinning, and it's exported in these huge sheets from Antofagasta, which is probably the ugliest town in the world. The one on the right is where we have the higher capex. We have, just back to this one, sorry. So we've got 12 square kilometres that need to be irrigated. We're in the middle of a desert. So what BHP Billiton does is they transport water 250 kilometres from a salt lake, and then they treat it with reverse osmosis to get the pure water. So you can imagine the cost that entails. With these biox plants, the same sort of thing. This is a gold biox plant in the goldfields of Western Australia, and this is a coal gold treatment plant in Uganda. They also require fresh water. In Western Australia, our artesian water has very high levels of total dissolved solids. It all has to be treated with reverse osmosis. So, this is, as I said, at least one reason why it has not been adopted as widely as it possibly could, because of the problem of needing fresh water. So work that was done very early on when we moved into this field was done by Carla Zammert, a previous PhD student of mine. And we were looking at, we already know that these organisms are inhibited by very low levels of chloride. So she looked at a range of microorganisms that are known to be iron sulfur oxidizing acidophiles, an increase in chloride levels, and she looked at the lag phase that was generated with the increase in chloride levels. And just to go through this reasonably quickly, so Acidothybacillus peroxidans is the workhorse of biomining research. It was the first identified. It was known that microbes were involved in the 1950s, even though this technology was used in Roman times. And we found, she showed that at as low as 0.6 grams per litre of chloride had a 48-hour lag phase in the oxidation of iron. And as you can see at the bottom there, seawater has 21 grams per litre of chloride. So we're not going to be able to use Acidothybacillus peroxidans, and it totally stops oxidising in this experiment of 4.2 grams per litre of chloride. Ferroplasma acidophyllum, leptospirophilum, and sulfobacillus sulfoxidans had a 24-hour lag phase at 4.2 grams per litre of chloride and gave up the ghost at 15 grams per litre of chloride. Pseudomicrobium peroxidans was able to tolerate up to 7.5 grams per litre of chloride, also gave up the ghost at 15. And the most tolerant one we had in our suite of organisms was Acidothybacillus cordus, which could tolerate up to 9 grams per litre of chloride, still well below seawater levels, and was unable to oxidise anymore up to 24 grams per litre of chloride. Now the reason for this is the mechanisms that acidophiles use to cope with their extreme environments. So an acidophile would be existing in an environment of say pH 1, and it's still able to maintain a circumneutral cytoplasmic pH. So we've got a delta pH of up to 5.5 units there. It does this through a range of mechanisms. One being that their membranes are a little bit more impermeable to protons. They have buffering capacity in their cytoplasm, active proton pumps, sequestering of protons. They have DNA repair mechanisms that don't deal with acidification. But the most important by far is the generation of reverse transmembrane potential. So bacteria generally have a positive external charge on their membranes and a negative on the inside of their membranes. These organisms are able to reverse that by pumping in potassium, and the positive internal charge repels the protons and enables it to maintain this delta pH. Now chloride is one of the very few anions that's able to pass through this membrane. So the chloride will pass through the membrane, negate that positive internal charge, lead to an influx of protons, acidification of the cytoplasm, and a reduction in proton motive force, and so a reduction in energy. So this leads us to my fascinating group of microorganisms, the Acidohalobacter, and it is able to cope with very high levels of chloride. Now there are 4 known species of this genus. The first one was identified by Huber and Stettner in the late 80s, and Paul Norris identified 2 more, all from the Aleutian Islands in the early 2000s. And then Carla Zammit, whose data I just gave to you earlier, isolated one in Western Australia a few years ago. Now we have a particular situation in Western Australia that makes us ideally suited to find these sorts of microorganisms due to the geology of our soils. So we have very acidic soils naturally because of a granitic base. We also, as you well know, have a lot of iron in Western Australia. But we have a particular problem called dryland salinity. So what has happened, land vegetation has been cleared for agriculture, leads to a rise of the water table, which dissolves the salts as it comes up, hits the surface, precipitates out, and makes the land non-arable. So what the farmers have been doing is they dig these channels in their paddocks, so the water doesn't actually hit the surface, it drains off before it hits the surface. But these channels are very low pH, high in iron and high in chloride, so a perfect environment to find these sorts of organisms, which we have. One we've called Acidohalobacter yilganensis because it was isolated from the Yilgarn crater. They are all mesophiles, they're all acidophiles, growing down to low as 1 for some of them. They're all able to oxidize iron up to above seawater levels. Their optimums are just a little bit below seawater levels, but they're able to cope at much higher levels. And most importantly for biomining, they're able to oxidize sulfide minerals in the presence of chloride, some of them up to 30. On the genetic basis, they're very similar, like the same sort of size genome, GC content. One of them has a plasmid, which we're yet to investigate, but it's quite interesting that it's there. I haven't gone into how they oxidize iron and sulfur. They all contain the same iron oxidizing system, but they have variations in the sulfur oxidizing systems that they use. And interestingly, the West Australian isolate is the only one that is able to tolerate copper and has the copper tolerance genes. They look very different under the microscope. My students don't believe me when they first start, but after a couple of years they work it out. Okay, so we wanted to, these actually had been originally named thioBacillus. They'd been, once 16S data was generated, Paul Norris identified they actually belong to the gamma protobacteriaceae, and then we have done further work on them and have shown to fit them within the chromatiales and renamed them Acidohalobacter. And we wanted to show, in demonstrating they're a separate genus, we wanted to see where they fitted into the chromatiales. The tree on the left is generated from concatenation of 30 ribosomal proteins, and the tree on the right from 28 universal proteins, and we have a very rigorous placement of these organisms within the order. So we're very confident we have 4 different species, and this has just recently been published in IJACM. So just to emphasise the difference, here we have thioBacillus peroxidans. I said salt tolerant, a sensitive one, sorry. We have generation of ferric from oxidation of ferrous with increasing chloride levels. I need to stand back a bit. We have at 0 grams a litre, it's very happy with its conversion of ferrous to ferric. By 10 grams a litre, it's really not very happy at all. unlike Acidohalobacter prosperus, where its optimum is at 20 grams a litre of chloride. It's very happy in the presence of chloride, and it's still oxidising iron very happily at 50 grams a litre of chloride. The scales, the index has fallen off. That's 60 grams a litre where it's actually had enough. It won't do it anymore. And as I said, most importantly, we're able to demonstrate it's still able to oxidise sulphide minerals in the presence of chloride. So up to 50 grams a litre for pentlandite, which is an acid-soluble mineral, and up to 30 grams a litre for pyrite, which is acid insoluble. So that was kind of good. Students tend to like to do a bit of genetic work. So we've started looking at the mechanisms of how are they actually to do this. So we know that they can do it, we know that we can apply them, we've actually applied them further on, which I can give you, not going to talk about today. We started looking at the mechanisms. So a PhD student, Himel Kalek, who finished late last year, did a genome comparison or genome analysis of the 4 species in this genome and identified the genes in all 4 species that will lead towards their ability to tolerate this osmotic stress or the high levels of chloride. And it's actually very similar to other halo-tolerant species. So the primary response that we see is that of the uptake of potassium and generation of amino acids and sugars to act as osmoprotectants. Then the secondary response takes over, where we have osmolytes produced, such as ectone, glycine, betaine and proline, as well as their transport mechanisms. So all the genes for these processes have been identified in all 4 genomes. And the specific chloride response that we see are a range of chloride ion pumps, channels, and also the outer membrane protein, which I'll go into a bit more in a minute. So we were drilling down further into this, and we looked at one of those species, the Acidohalobacter alionis, for their protein expression or protein abundance. We used SWAF for that, and we're able to identify over 2,500 proteins, of which 600 were differentially abundant. And we compared the abundance of proteins for the sensitive strain, Acidobacillus peroxidans, at 0 and 6 grams a litre, and Acidohalobacter alionis at 5, 15 and 30 grams a litre. What I just want to take from this heat map is that you can see that there's not a difference, there's no difference in abundance, or very little difference in abundance, with stressed and non-stressed and stressed for Acidobacillus peroxidans, whereas we're seeing a very different abundance of these proteins as the stress increases for Acidohalobacter alionis. So it has a very osmotic or chloride dependent stress response. And just to emphasise this, the proteins that are produced by Acidobacillus peroxidans at 0 grams a litre also cluster with those produced at 6 grams a litre, so no difference. Whereas the response of the Acidohalobacter alionis at 5, 15 and 30 all cluster very separately. So we're getting a very distinct response in, to this osmotic stress or the chloride stress. Just as a quick snapshot, as I said, we've got 600 differentially expressed proteins, and I won't make you sit through all of those, but just to pick the main ones of interest, we saw over a 400-fold increase in the proteins involved in the production of the osmolyte ectone. Now, ectone has not been identified in any other acidophiles, but it's commonly seen in halophiles or halotolerant microorganisms. Other osmoprotectants such as glutamine and glutathione also we see increased protein abundance for those. We see, not surprisingly, increased abundance of proteins involved in cell and membrane integrity. It's undergoing stress, you would expect to see that. And we also see an increased abundance in proteins involved in DNA repair and protein folding. Once again, not that surprising. We do see a decrease in the proteins involved in carbon fixation. All the enzymes involved in the Calvin-Bassen-Benson cycle are reduced in abundance. We have actually done some protein work on other species. We also saw that there too. We also see a decrease in the production of the outer membrane protein, and we believe that that is probably due to preventing or reducing the flow of chloride into the cell. Interestingly, we have seen a reduction in some of the proteins involved in iron and sulfur oxidation, which does seem counterintuitive. It's not all of the enzymes, not all of the proteins involved in these processes, and we need to interrogate that further. So Himel loves drawing models, so I promised I'd put this one up. So This was published in Research in Microbiology, and so we see an increase in pumps of iron pumps. We see an increase in the proteins involved in membrane integrity, and we see an increase in production of osmoprotectants as well as DNA repair mechanisms. We see a reduction in the carbon enzymes involved in carbon fixation, the outer membrane protein, and interestingly, iron and sulfur oxidation of some of the proteins as well. One last thing I'd like to leave you with. I have a new PhD student starting at the end of the month who will be working on this, but the question we have is Acidohalobacter a halophile that has learned to deal with acidic conditions, or is it an acidophile that has learned to deal with halophilic conditions? Our gut feeling is it started off as a halophile. The ectone is the big key for us. Ectone is only found in halophiles, it's not found in acidophiles. So we suspect that the genes for salt tolerance have been inherited vertically, whereas the genes for acid tolerance have been inherited through lateral gene transfer. But watch this space. Hopefully we'll have an answer for you in that very soon. And finally, I would just like to thank the people involved, particularly the PhD students Hamel Kalek, who's done the genome and the protein work, and Carla Zammit, who started the ball rolling on this with the original physiological work as well as the isolation of the organism. There's been a range of honours students that have worked on this, so Frankie, Tim and Kieran, and Melissa Corbett was a PhD student who's done a lot of work on the bioleaching, which I haven't presented here. The early work was done in collaboration with Mark Dobson from the Linnaeus University in Sweden, and the genomic work was done in collaboration with David Holmes's group in Santiago in the Centre of Bioinformatics and Genome Biology, and particularly his postdoc, Carolina Gonzalez, has been invaluable in this. And then Paul Norris, he was at Warwick, he's now at the University of Exeter. He donated 2 of the strains to the study and the various funding bodies. Thanks very much.
Thank you, Liz. Do you have a question? Andy, you want to kick it off again?
Really short. Given the spectrum of halotolerance across the strains that you were looking at, does that reflect where you might have some transporters for the osmoprotectants at the lower end only, and transporters plus the biocontrol pathways of the extreme ones?
I don't know. Well,
are they piggybacking on someone else's ability?
Well, there is some work, we haven't done the extension work on this, this is all field project work. There was some work from Giovanna's group where they looked at Iron Mountain, which is where she's had a lot of the work, and they did find that they didn't find, it's not this sort of organism, they found that there's a piggyback between leptospirillum and some of the other organisms where fungi, for example, producing chlorine and leptospirillum is taken out. But that was all field culture work. They don't produce the same as replicator cells.
I got a quick one. Thinking about field application, I know challenges thinking about taking non-resilient organisms to Brazil. Is that the same in biology? Would you be able to apply this in Chile? These salt tolerant Western Australian organisms in biology?
It is also a problem, isn't it?
Is it for biology?
Well, it depends which biotechnology process you're using. So in bioxidation plants, they do inoculate. In those dump species, they just speak to nature. So whatever's present. Same problem. So any work. So we have done some pollen work. We had the same problem issue. The inoculant strains disappear. From other work that we're doing in a different system altogether, I'm really coming to the theory that the inoculant strains just kickstart things and then promote an environment for any indigenous microorganisms to take over. Now that's coming from a totally different system with phosphate solubilizing bacteria and rare earth elements, but I think it could be universally true. It just needs to be, sometimes it just needs to be given a bit of a starter to get going. I suspect that's not the same with yours, and that's what happened in our system. That doesn't answer your question. It's coming to Australia in no way with our quarantine regulations.
So you had optimal growth at 20 grams per litre.
Yeah.
Lower than that was problematic?
Yes, and well, we have shown that 3 that came from Italy have an absolute requirement for the presence of chloride. We don't know exactly what fluoride's doing yet, but it does have an absolute requirement. The one from Western Australia is able to grow in the absence of fluoride, but it just doesn't like it very much. So we actually think, and I haven't presented the data here, but we think that the one from Western Australia is actually more of an osmophile than a halophile. So we have done work where we've grown it. It does have a requirement for fluoride in that case, but we've been able to grow it at very low levels of fluoride, but with magnesium sulfate instead to give it the osmotic stress.
And does it make a difference if your fluoride challenge comes from sodium fluoride versus potassium fluoride?
Haven't tried with potassium fluoride. We haven't used potassium fluoride because of the potassium. Good question. The experiments we've done doesn't match it on where the form of fluoride comes.
Any further questions? Going on? It's good time. All right, last speaker. Who was that again? Oh yes. So introducing Emma was like introducing my twin. Introducing Liz was like introducing Florence the Machine. Introducing Jim Teege is like introducing someone from a superior civilization from a planet far, far away. It's my great pleasure to introduce to you Jim Teege, a young, up-and-coming microbiologist. He has degrees from Iowa State and Cornell University. He's now a distinguished professor of microbiology and molecular genetics and of plant, soil, and microbial sciences, and director at the Center of Microbial Ecology at Michigan State University, home of the Ribosomal Database Project. Get pens and papers out, write these numbers down, it's quite extraordinary. So he has over 500 referee publications. 12 of those have over 1,000 citations. He has one publication with over 8,000 citations. 7 of these publications are in Science or Nature. He has an H index of 153. And has amassed over his extraordinary career a total citation body. So his work, published work, has been cited just shy of 100,000 times. It's around 96,000 times. Really quite remarkable. Jim's also a member of the US National Academy of Sciences, amongst other associations, who's also a past president of, not the United States, but the American Society for Microbiology and of the International Society for Microbial Ecology, for which we owe a debt of gratitude because they're a regular sponsor. Jim's had crucial roles in establishing the International Society for Microbial Ecology and the ISB Journal. He has made major contributions to microbial ecology and physiology and diversity, especially in relation to nitrogen cycling. I think his major contributions are there. But he's also been, and this is what I know him for, a pioneer riding the crest of the wave of technology developments over the last 3 decades, and he's really stayed abreast of all these contemporary molecular methods in microbial ecology and been a pioneer developing methods and applying them, promoting them through all sorts of different phases, through our ugly fingerprinting phase to our now more elegant sequencing phase. And I mentioned the Ribosomal Database Project as well. It comes out of his group. But for me, sort of on a personal note, for me personally, the most important thing Jim Teege has done is publish a paper in Science in 1982, when I was 9 years old, which was also first authored by a young scientist at the time named Joseph Saflita. Was he postdocing with you, Jim?
Yes.
Yeah, postdocing with you, who I have enormous respect for as well. This paper was entitled Dehalogenation, a Novel Pathway for the Anaerobic Biodegradation of Haloaromatic Compounds. For those of you who know the work that I do, Matt Lee and my group and a bunch of my students, we all work on reductive dechlorination, and he kicked it off. So it's with great, great pleasure to introduce to you Jim Teege.
Thanks, Mike, and it's a pleasure to be here. I don't know where he got all those statistics. I don't know those statistics about my background. But one statistic is, look at my shirt. Does anybody have one like that?
No.
Anybody here designed this shirt? designed this was a microbiology graduate student who won the award for the design at the Australian Microbiology meeting in 1992. So you have to be about 50 years old.
I spoke at that meeting.
So I don't get a chance to wear this too often, but it's a pretty good shirt. And it was sponsored by Delta West Airlines. Anybody fly Delta West? Okay, so that's a little history. So I've been to Australia a number of times, but I haven't been to Sydney for 20 years. And the last time I was here, they were building buildings day and night for the Olympics, and I could hardly sleep because they were counting the time. Okay, so I'm going to talk a little bit about the microbiomes and about their status and future. Okay, so why is microbial ecology important? Because microbes do a number of things, and a lot of times it relates to money. So I put that in the context. They cause problems, which costs money. They make money, and they sustain our environment, which saves money. So from a public and a political perspective, sometimes it's important to say how microbes are of value. And these involve communities, and that's microbiomes. This figure also has some history. So Joe Handelsman and I chaired this committee for the National Research Council that published this report in 2007 on the new science of metagenomics. And I drew this figure at that period of time. And I thought the field would expand. And at that time it was all about sequencing. That was even before, certainly before Illumina came on the market. But looking ahead, it was the analysis of that sequencing that I was worried about. How could we handle that? Would be on top of that. But then we would come in this experimental phase where we would do some major hypothesis testing. So now that was more than 10 years ago. Now we're there. So now we need a new figure. And I usually challenge people to do their figure for the next 10 years. And I think we're in a stage of speciation. In other words, this breaks down into what is important for different habitats. So if you're in the marine environment, or you're in waste treatment, or you're in bioleaching, you would draw a different figure. And it's important to think about the future and then how you might draw that figure. This has some history. The first time I showed this figure was at the ISME meetings in Cairns in 2006. So it's back in Australia. So my outline is this. So I'll talk about two examples of metagenomics. And first I'll give my perspective, or at least my view on genetic complexity of microbiomes. And I generally work in the soil environment, but I've worked in other environments as well. And the figure on the right-hand side in particular is the phylotypes in only 3 grams of Midwest US soil from 3 different states. And you see a distribution here that you would expect for ecological variation. But the importance is the diversity over 4 orders of magnitude in that small amount of soil. So at an organismal level, diversity is really high in the soil environment. If we look at the genetic environment, this was done by a former student, Kostas Konstantinidis, who published this technique to evaluate the genetic diversity, or basically sequence diversity, in microbiomes. And this is that first paper in which it was acid mine drainage, human intestinal tract, freshwater lakes, and soil. And the Midwest soil runs around 0.61 terabase requirement for covering most of the genetic diversity in those samples. And then carrying that forward, developing new computational methods to get at these habitats that are more complex. We took 86 metagenomes and evaluated their metagenomic diversity. And at the bottom axis, we can come up with an alpha diversity for that metagenome. as you can see across here. And it relates to 16S diversity as well for these various different habitats. But as we go in this direction, we get into the external environments, and particularly the soil environments. So genetic diversity is really high. So how are we going to deal with that? And how can we move forward to understand organisms better? And again, working with Costas, we together have gotten grants from the National Science Foundation to try to move forward in evaluating diversity below the species, or below the genus level. 16S works great at genus and above, but we're not so good at the lower level. And so how we might do that. And this then was developed by using these techniques that Costas started with. He was in my lab with average nucleotide identity, average amino acid identity, and then scaling down, searching and scaling down until one gets down to a finer level of resolution. And then what this leads to is further resolution of particular organisms. So this information now is mostly from pathogens, because that's where we have a large number of gene sequences to evaluate that kind of information. So this is Campylobacter junii with a species cutoff of 95% ANI, which seems to hold pretty well for general species definition. But it recognizes then 32 clades within that. So there is structure within that species. And then that will allow us to get to a finer level of resolution, which can be important for ecological distinctions. We can also use this information then to evaluate the pangenome and the core genome. So I consider this sort of the genetic image of a species, this being the genes in common in all of the sequenced organisms, and you get down to not very many that are in common in all members of that species. And this represents the pangenome, which is the genetic diversity within that species. Okay, so that's a look into the future, where we would go with regard to evaluating organisms at a finer level of resolution. So I'm advocating moving beyond 16S. Actually, at a meeting the American Academy of Microbiology had in 1996, I said, we need to get beyond 16S because that's the level of resolution we need for ecological understanding. But 16S is great. It's like measuring the pH of a particular sample. We always need it, but we need more than that. That's the big one. So this is an ecological framework for looking at community outcomes. Okay, so what structures a community? So some of it is deterministic, and of course some of it is stochastic. And these, of course, would be the major drivers that determine the outcome of that community, and it primarily reflects the dynamics of that community and how strong that deterministic selection is. But in the microbial world, someone's always going to be stochastic. So questions that we like to answer would be, what features are deterministic? How much is deterministic? How much of the unexplained is the unmeasured, which we often have a problem with because we're not sure what, and sometimes we can't measure what might be important drivers. How much is due to non-uniform inoculation or dispersal? And is system dynamics the primary determinant of this distribution? These are all questions that relate to community outcomes. So an important goal then is how to herd the community structure for the desired outcomes in many of our applications. So in the omics era, the goal is to link activity to function, because it's function that usually pays the bill. And in this omics era that we've been in now for more than a decade, sometimes we lose track of getting a good handle on function. So metagenomics is DNA based, and it is of value because if the concentration of DNA is high, it means the organism grew. It's a long-term measure of the outcome of growth. So that's a value. We can have metagenomics also, about RNA. And in many cases, that's a very short-term activity measure, sometimes too short to be useful. So one is too long and the other is too short. We can go to proteomics, and that's the catalyst. That's somewhere in between, generally more stable. But all of these require the presence of substrate and compatible conditions for activity, which we don't always know. We've got metabolomics, where we have a product, and we can measure rate of change, but in a lot of systems, that's not specific for which genotype. So metabolomics is a community outcome where these measures are the specific organisms responsible. Now, there's certainly progress been made in being able to link omics to activity for particular habitats, but this still is a grand challenge. In some cases, perhaps an intractable problem. So in my outline, then a couple examples from metagenomics. So using metagenomics to interrogate the rhizosphere of biofuel crops. So in the US, we've had over 10 years of large DOE funding for bioenergy projects. And the projects at University of Wisconsin and Michigan State had a component that was focused on the sustainability of this biofuel economy, which included biogeochemistry, low-cost production, economics, social effects, those kinds of things. And we focused on corn, switchgrass, and miscanthus. And an important part of the bioenergy sustainability is that 40% of energy production is projected to be nitrogen fertilizer. So you want to minimize that. So that means the nitrogen cycle, understanding that nitrogen cycle and how those systems might be able to minimize the loss of nitrogen. So we developed a gene-targeted assembly approach so we could target those genes and assemble those, and then evaluate those in the rhizosphere of this crop. Now, switchgrass is a primary crop of interest. It's native to most of the central and eastern U.S. with ecotypes for particular subregions. It's a summer active grass and quite productive. And it is not nitrogen responsive. So this is 8 years of a fertilizer addition trial. So this is the amount of fertilizer added across the bottom. This was the first year the plot was set up. These were the additional 4 years, and these are the subsequent 4 years. And this is year 8. And you see, after no fertilizer input for 8 years, high productivity, still no yield response to nitrogen fertilizer. So the nitrogen deficit over that period of time is about 50 kilograms of nitrogen per hectare per year. So how is that happening? Where is the nitrogen going? So presumably nitrogen fixation. Now, if we look at the rhizosphere of this plant.
Good afternoon. We would like to remind you that the museum will be closing at 5 o'clock today. Please take this opportunity to complete your viewing or perhaps visit our shop located on the ground floor. Repeating, the museum will be closing at 5 o'clock today. Please take this opportunity to complete your viewing or visit our shop located on the ground floor.
Okay, I'll complete my talk. Okay, so this is NIFH, archaeal nitrifiers, bacterial nitrifiers, denitrification pathway is illustrated here. And this is the quantity of those genes normalized to RPLB, which is 1 copy per cell. So you can see the quantities of those different genes, and then this is the sequence matched to the nearest taxa in the database. And these are the 3 crops: corn, miscanthus, and switchgrass. So not huge difference between the different crops. DIFH is about 1 per every 300 cells. The denitrifier genes are much higher concentrations. And if we look particularly at NIFH in the different crops, so this is corn. We have 7 replicates as illustrated here. And these are the taxa that are selected by those crops. So the interesting thing is the corn is selecting rhizobium and bradyrhizobium in in its rhizosphere. Piscanthus is selecting an organism whose nearest match is this marine nitrogen mixer, but the sequence is only 70% similar to this organism. Obviously, there's another organism there, not yet isolated in sequence, that's in that habitat. And switchlet grass is selecting a different type. This Novus pneumonium is illustrated here. So while it looks like the 7 replicants are variable, there are certainly patterns that are consistent with each crop. So nitrogen fixation is selected in the rhizosphere. Now, in recent work that Sarah Raleigh, who is an ecosystem ecologist, did, tried to look at measuring nitrogen fixation directly in these plants, in the leaves, in the roots, presumably from nitrogen fixation, but not accounted for by other sources in these 15 gas and a conserved tracer into the field, and then using that ratio to calculate diffusion and the amount fixed in the plant. So she could measure some episodic nitrogen fixation using that system. So a big point here is that these grasses, I think, do fix some nitrogen. In an experiment I did when I was in Australia with Gupta five years ago, also on grasses in that area, and this was 3 different grasses, again measuring nitrogen fixation. It's illustrated here in the roots and in the rhizosphere soil for these different grasses. Also measuring in plant parts, in the leaves and the roots. And then we looked for the NIF-H genes in the plant material, both in the Michigan biofuel crops and these Australian crops, and similar, except in the soil case here in Michigan, all of the other nearest taxa were similar. So the big point is that I think there's certainly nitrogen fixation going on associated with these systems, these stress nitrogen systems, that is beneficial in natural ecosystems. Now the last one. One of the things that we focused on in the last several years is what I call eco-functional genes. In other words, genes that directly control important ecological functions. What determines their activity, how they are distributed in different systems, and who are the responsible organisms. So one of those is antibiotic resistance genes. And we already had a nice introduction to that topic today. And I'll give my view on that. So this is a paper published by Guangzhou out of Guangzhou in China, showing the amount of antibiotic use. On this axis is daily fine dose for 1,000 habitats. So comparing China, UK, and US. And this is the antibiotic use in tons in the US, UK, and China on the same scale, with US and UK blown up here and distributed according to that use in animals and humans. The point is here, the point he was making is that there are lots of antibiotics used in China, and generally in Southeast Asia overall, lots are used. So we do have a strong selection problem going on in the environment globally. The One Health concept was mentioned earlier, which is a really important one. It includes the animal, and up here in the animal I put livestock, aquaculture, and wildlife. And in the environment, which is soil, water, and air, and the human, and I put pets here because the exchange between humans and pets can be quite significant and different from the dynamics with these other animals. Important point in this figure then is the paths of transfer of the antibiotics and the arrowheads, because the direction can be in both ways. It's often difficult to resolve, and that's probably important to understand the problem. So between humans and animals, which is the direction here? And it may be different in different settings, but to not assume that it's the animals that contribute to the humans or vice versa, it would be inappropriate. So what do we know about the natural resistome? And as mentioned earlier, the antibiotic resistance came from the environment. The antibiotics came from the environment. Many of those organisms are resistant to those antibiotics. So most antibiotics are from soil microbes. Many soil bacteria contain functional ARBs. Pseudomonas, Burkholderia, they may have 6, 10, 12 resistances already in them from just initial soil isolation. Certain ARG sequences are found almost everywhere, and I'll say more about that. Some ARGs have alternate functions that may dominate their selection history. What we don't know is how much of the selection we see is for the microbe and not for the ARG. How much of the ARGs are functional that we detect from a molecular point of view, and how much of the ARGs are mobile elements, because that's where the risk really resides when they're mobile. So lots of things we don't know, and it's difficult because of the background problem. Now, this is a study that Bill Hoban and I did like 30 years ago, where we screened soils from different parts of the world for resistance to antibiotics. So this is the percent of resistant on this side. So if it's at 100%, that means the plate count showed no reduction in the presence of the antibiotic. So for this first antibiotic, there was only a small reduction in the plate count of soil. These are lined up in a rank order, and the general pattern is pretty much common globally. I mean, there are some that are sort of outliers here, but that's what you would expect for background level of functional resistance in soils from most parts of the world. The omics methods that we use for assessing ARGs are illustrated here. So we use highly parallel qPCR. So we used what was originally the Wafergen system, 384 primer pairs, 5,184 assays per chip. Based on work on over 300 chips from different environmental samples, we were able to update those primer pairs. So that's primer set 2.0, which was published last year, for both ARGs and mobile genetic elements, so that we can screen for both. The advantage of this kind of system is it's more sensitive, and you can do a lot of samples with this number of assays per chip. We also do gene finding in metagenomics, shotgun data. There are several databases now available to facilitate that. We use the SAR database, which is Structured Antibiotic Resistant Gene Database, which has structured according to gene type, and the subtypes within that gene set based on HMM models with 80% aligned identity being the cutoff. It has the advantage of no primer bias, and of course it also recovers some non-specific conserved regions as well. And then we've done some work with long read sequencing, both PacBio and Nanopore. The advantage is you get the full genes, and you get their linkages and if linked mobile elements. This probably has the greatest potential in the future for understanding this and other biological problems, but the DNA quality is that's really a demanding aspect of this, and the error rate is still a challenge. So this is one study we did with the Chinese collaborators, and the big thing here was that this was a study in controlled plots for other purposes, but over 26 years. And these plots were both aerobic agriculture production for peanuts, in this case dry land, and also paddy rice. So two different ecological systems, one aerobic and one anaerobic. These are using this highly parallel qPCR. These are the different resistant genes that we assayed here. This is the manure that went on the soil. So you can see that there was resistance in that manure. Then there are controls which no fertilizer was supplied, and controls with just chemical fertilizer and then the manure as a fertilizer. If there's an M here, it means it was the sample with manure. Dry land, manure, and wetland manure here. But the big point here is that you don't see that the gene detection level is low to 0, and you don't see much difference in the manure samples versus the non-manure samples. Hence, there's a no buildup of ARCs from long-term manure fertilization. And the ARCs that are detected are presumably the natural rhizostome, and because they're also found in the non-manured soil. So the big point here is that the standard practice of manures that are containing antibiotics, and I would also say that we assay a year after the manure was added to the soil for antibiotics, and we can detect 6 antibiotics still in the soil in these soils here 1 year after the previous manure application. So selection was there, but yet the ARCs are not selected after a period of time. Now looking at the metagenomic data, so in this case we're looking at some native soils. So we have metagenomes from 26 soils, and they are from the Alaska tundra below the subsurface, so these clearly are not influenced by human activity. U.S. Midwest prairie agriculture, and here we have paired native prairie and 100 years of cultivation, and Amazon rainforest paired with the same rainforest area that has now been converted to I think about 20 years of cattle grazing. So the biggest land use changes are cultivated agriculture, and in the tropics then conversion to pasture land from rainforest. So what do those two kinds of management changes do on the ARC system? So here we detected 240 ARC subtypes. 55 of those ARCs we consider background ARCs because they were found in all 26 samples. And 81 to 98%, that's in the Alaska case, of the detected ARCs are part of this global background. In terms of abundant, this is what they are. So the majority are EFOP spuns, and of course they're there for other reasons. And there's a lot of regulator genes also in terms of abundance. If we look at the diversity of the different types, this is cell protection, and this is deactivation. Their quantities are much lower in terms of total abundance. So that gives an idea of what this background is in nature. If we look at particular resistances, as illustrated over here, in the native and managed soil. So this would be the Alaska case. This would be the Midwest native soil, and it's paired 100-year cultivated soil. Not much difference. In fact, there's no statistical difference between these two. This is the Amazon rainforest and the Amazon pasture. In this case, the conversion changed statistically. 6 of the ARCs were enriched in the pasture conversion. So an overview of how that land use change has affected things. Now, an important point is whether these ARCs in nature are clinically important ARCs, and how similar they are to those. So this is evaluating, I show an example here of 8. We took 12 clinically important ARCs that are more directly controlled resistance. And the blue bar is the total detected by the same method that we used before. And then the colors represent how similar the amino acid sequence is to the clinical ARC. So as we go in this direction, the last red here is 97% identical in amino acid sequence to the clinical ARC. So you see in some cases, they're very dissimilar from the known clinical ARC. In others, some are more similar in getting close.
Good afternoon. The museum is now
none are
closing.
identical.
Please make your way down to the ground floor to exit via Crystal Hall. Repeating, The museum is now closing. Please make your way down to the ground floor to exit via Crystal Hall. We hope you enjoyed your visit to the Australian Museum and look forward to seeing you again very soon.
Yeah, we got it. Okay, so this is the long read. So we want to do the long read, and this was done with dairy cattle. They're treated with cefaclor. We took the manure, we enriched it a bit in advance, and then we did PacBio and nanopore sequencing. The summary point here is that the outcome from both of those technologies was about the same. So in this case, it evaluates whether we got the complete gene. So in most cases, both cases, we got about the same amount of the complete gene, 70 and 67% here. But the big question is, can we see linked ARGs, and are they on mobile elements? So the red bar here is the number of sequences that we obtain for particular functions. So for metal resistance here at the top, lots of metal resistance. But the blue is whether it was connected to a mobile genetic element. Very few. Multi-drug resistance, almost none connected. Only 1% connected to a mobile element. Biocide is also very low. And if we go down here, we see for some of the others, aminoglycosides, 25. Here's tetracycline, beta-lactamases, sulfonylamide. So some of these others have the numbers of those genes are much less, but they are linked to ours. So an important point in the long range is be able to assess these actual linkages and if they're on mobile elements. And this will be an example of one of those, which you see the mobile element, and then the different kinds of resistances that are on that mobile element. So it would suggest that co-selection is alive and working as one might expect. And to finish off here, what's for the future? So my colleague, Syed Hasham, has then developed this handheld device, illustrated here. It can have up to 64 wells, isothermal amplification, can be run by an iPhone, can send data, receive data. So it's basically a cheap lab on a chip. And we use the integrase gene as an example, illustrated here. And this important part here is that this was done by high school students and community college undergraduate students, local community college, from samples taken different places in Michigan and also in Korea. And this is what the students got here. And this is a regular qPCR assay. So pretty good correlation. So the point is that in the future, why not? We can do things like this on-site very reasonable. And to finish off, what about the future? So these last few slides are going to be the ones I gave at ISME. So if you were at ISME, you heard this. And I said that our field has been technology enabled. And Andrew pointed out that it's more than technology, and I would agree. It's the questions that we use with the technology. When technologies come and are advantageous, we will use them. It's the questions that we ask that become important. So if I look over my era, it would, at first, all we do is measure functions and processes. We did incubations, we did isotopes, we did gasless chromatography, we had allo analyzers. We were measuring the processes. We knew less about the organism, but we could measure the process. And then in the 80s came the PCs. Then we could do kinetics, and we could do math models. In the 90s, we could sequence genes. I mean, one PhD thesis for a gene. And we could do a DNA extraction. We could do hybridization. 16S came. Then the internet came. In the 2000s, we could do whole genome sequences. And we could do high throughput sequencing and assembly. And in the 2010s, the era we're just finishing, we can do metagenomics, transcriptomics, proteomics, low cost through high sequencing, high performing computer centers. And what's next? What's next? Okay, so here's my thoughts. For the next 5 years, that's easier. Return to the questions of the 1970s and 80s. more process measures, more modeling, more culture-based resolution, but with more resolving methods. We can do long-read sequencing technology, that will become standard. Use of advanced microscopies at the microbe scale. Image analysis, that's very computationally compatible. More use of isotopic methods, linking to function. Better habitat characterization and its reporting. We don't do a good job of that. SAGs and MAGs become part of organized taxonomy. Use whole genome information, not only 16S. And meta-analysis of the decade of diversity. So, next 5 years, that's my predictions. And then the longer-term future. What about that? In-field, rapid, e-connected diagnostics, artificial intelligence guidance for image analysis, enhanced metabolite and chemical analysis, multiple omics markers. So I would expect that to come forward. High throughput culturing, ecological, physiological, and biochemical characterizations. We've got to get back to the live organism. And we just saw a talk where understanding the live organism is really important. DNA markers for kinetic properties. We can understand competition by predicting its kinetic capabilities. Real-time E-link omics with function. CRISPR-Cas directed microbial design. Particular microbiome and management ownership. Synthetic ecobiology. Designer communities. Quantum computing, complex system, complex solutions. Okay, so that's my thoughts for the next 20 years or beyond. Now, how good will they be? Who knows? But in this last slide, I'll say probably it's difficult. So Tom Watson, the founder of IBM, said there will be a world market for 85 computers. Now these guys are all successful. They didn't predict the future very well, but they're so successful. Ken Olsen, founder of Digital, said there's no reason anyone would want a computer in their home. And Bill Gates said 640K ought to be enough for anybody. So, predicting the future is hard. What is really hard is the game changers. You know, who could predict the internet and the impact that it's going to have, that it had, or cell phones, or all of that. What we've done, we've been able to store, process, and transport electrons at a fine scale. So if we think about that, maybe that helps us for the next era. The problem is never how to get new ideas into your mind. It's how to get old ideas out. So students, you have an advantage. You don't have all these old ideas in your mind. Maybe at your thesis defense you need them, but maybe that's all. Okay, so that's my thoughts about the future. And of course, many people at my lab over the last decade and beyond have contributed to these different projects as indicated here. So thanks.
While he's here, anybody got any questions? Everyone just wants to be here.
Yeah.
Somebody's got to ask one. Thanks, Jim. When you talked about linking the various resistance genes to mobile elements with the PacBio sequencing, how exactly did you make that linkage? Were the reads long enough that you were looking for a specific
Yes. Yes. Because the way we prepared the nanopore, we didn't go for the longest reads. We had to cut them in smaller sections, but then we can easily assemble and get the
Following up from Andy's question, because there's so many diverse mobile genetic elements, like how did you define what were mobile elements? Like what are you looking for? Like do you think that you captured all the mobile elements? Like what did you use as
Did we catch all of them?
Yeah.
In an example? No.
Yeah.
No.
Because it's very hard to define the end of mobile elements.
Right.
How large they are. And how did you try to combat that? How did you try to capture everything?
I wouldn't say we tried. We had long reads, and we determined what was on the read. Yeah. So in terms of comprehensive, no. Because we also had short pieces too that would have information on them that we didn't bother with analyzing them.
Jim, I've got a broader question. You began the talk looking at some ecological theory about how much of community assembly is deterministic and how much is stochastic. How closer in your career do you think we've got to answering that question?
I think we've gotten farther away from it. And actually, I just was involved in doing a paper with Jisong Zhao on this because he's been very interested in this. And it's a very mathematical paper that deals with simulated data as well as some experimental data. And basically the outcome of that says that and we did certain things like scale and ecological, environmental noise and things like that. At scale, it doesn't work very well at all. And the accuracy, you can see particular trends, but it's still not very accurate. The paper is going to provide some guidance. Basically, it's a null model based approach. Provide some guidance, but it leads me to my first answer is that this is really hard.
Yeah.
And we'll be at it for a while.
Yeah, keeping us busy. All right, are we all out of questions? Oh, Sally?
Just a little one. When you had the cultivated and uncultivated soils, and they were very similar resistance profiles, how did that link to just the community structure? Were you in any way able to predict? Because one would assume if it isn't human impacts, it would be purely determined by what species were there, but you would expect the species to be shifted by cultivation. Were you able to predict or link those two?
Like in the Midwest soils, we had done a previous study, extensive 16S sequencing, and those communities are shifted.
Yep.
Not in a huge way, but you can definitely see the difference in them. But at the ARG level, we don't see that shift. But in none that I showed here, but in some particular samples where we do see a shift in the community, we can link that to the ARGs being shifted with them. So the ARGs are being carried along by the ecological selection.
Yeah.
Yeah. I think in general that's a pretty common.
Aaron?
Another question about the ARGs and mobile genetic elements for the long reads. Does the association with the mobile genetic element say something about how recently the particular gene was acquired, and also in turn then something about new selective forces that might be acting on these microbes?
Yes. My student has a different opinion about that than me. He thinks that recent selection was an important part, because also part of what to get this to work, we did a selection of the manure in the presence of the antibiotics first. So we got better quality DNA and probably enriched it. He thinks that also contributed to some further selection. I'm not sure in that period of time it could have happened. But this will be very useful for those kind of questions.
So Jim's going to come to the pub with us, so we might save further questions for that. We thank you, Jim, for being here. I'll hand over to Yolanda for some final announcements and wrap up.
Wonderful. That concludes the formal part of our proceedings this afternoon. Please join me again in thanking all of our speakers, sponsors and volunteers for today. presented a poster today. I believe they've all been brought down to the front here with the canisters as well, so just make sure you handle everybody else's posters carefully. Dinner will kick off at 6 p.m. upstairs at Harpoon Harry's, which is shown on page 4 of the program. It's about a 10-minute walk from here. Dinner will be served from 6:30 p.m. There is a bar tab which commences at 6 p.m. and runs until 9 p.m. or until it runs out. So I assume it's going to last until 9 p.m. Importantly, we do have a number of treats for you tonight. So upon arrival at Harpoon Harry, you will receive a complimentary cocktail thanks to our sponsor 4 Pillars, who have created 2 specialty cocktails for JAMS attendees tonight. 1 gram-positive cocktail, 1 gram-negative cocktail. You get to choose which one you prefer. Aside from this, we have 3 more specialty cocktails created especially for tonight. The Illumina cocktail, the In Vitro cocktail, and the new JAMS signature cocktail. Ingredients for each drink will be available at Harpoon Harry, so you know what you're actually drinking. Once again, thank you for joining us at JAMS 8.